Morning, grade 9. Last class we saw about lysosome. What is the function of lysosome? And we covered all those cell organelles and its function. And next we want to see the structure of microscope. Today I'll give some idea about microscope. After the reopening, with the help of a original microscope, we'll see all those parts. Okay. So this is the structure of one compound microscope or light microscope. Why we or call it as light microscope means here the source is light. So we are using light to see the object. So we'll call it as a light microscope. And why we are call it as compound microscope means normally we know some hand microscope, that is simple microscope. We can able to maximize the objects in their 5x. We can see the objects into 5x only we can able to see in the simple microscope. And in this compound microscope we can see maximum 40x or 50x, like that we can able to multiply we don't want to multiply the microscope itself, it will multiply and it will show the objects. So we can able to see clear objects in the microscope. So what is this microscope? Nature of the microscope is compound microscope or light microscope. And these are the structure, these are the parts of the microscope. The first main part, this is eyepiece. So this we can able to remove also, we can able to again replace it. With that only we'll keep our eyes to see the object. So that is we used to call eyepiece. Okay, that is we used to call eyepiece. And here we have a knob. Okay, here we have a knob that we'll call it as coarse adjustment. So we can able to adjust to get a fine or to get a clear picture, that we'll call it as coarse adjustment. Even fine adjustment, this is also to get a view, clear cut view. And here we can able to hold this microscope with the help of arm. Okay, arm as a structure. And the straight tube is called body tube. Okay, straight tube we'll call it as body tube. Next here we have a two area also we can see objective lens. Okay, here one lens, here one lens. So that this objective lens only will magnify our objects and we can able to see our clear view of the objects. And here a stage. Okay, stage means here this is a stage, there only we can able to keep our slide. Okay, and here down we can see mirror. So the mirror is only it's getting light and will help, with the help of the light only we can able to see the object. And down we call it as base. And below to the stage we can see condenser. It will help to condense the light so we can able to see the objects clearly. And here it was given microscope slide. Slide means a small rectangular glass. Okay, that will call it a slide. With that we'll take a section and we'll keep it in the slide and we can keep it in the stage and we can able to see the objects with the help of the eyepiece. That's all about the structure of microscope. You just draw and practice in home the structure of microscope and label the parts. It was clearly given in your NCERT book. After the reopening we can see with the help of the true microscope, and I'll demo some with the help of some slides. Thank you, children. Next we'll see the very interesting a process called osmosis. So we have covered structure of microscope, what is structure of plant cell, animal cells, difference between plant and animal cells, and what are the cell organelles which is present in plant cell, animal cells, what are those structure, what are the functions. I think you people are clear about all those things. And next very important interesting topic, we'll call it as osmosis. Okay, osmosis is a process which will happen between cells. Okay, between cells. Because of this osmosis only, a movement of a material or movement of a liquid from one cell to another cells will takes place. So we want to see in a detail. So we'll tell this is the role of semi-permeable membrane or plasma membrane. So we can write whatever I am giving the points, all those you can write under the plasma membrane. Why? We know the plasma membrane, through that plasma membrane only all those material will pass, that is it will get inside to the cells and get out from the cells. So whatever I am giving you can write under the role of plasma membrane also, or separately the Sahodaya will ask questions. What is osmosis, types of osmosis, and also types of solution. Everything I am going to narrate today. Clear, children? So we'll see there are two types of osmosis. One is endosmosis, another one is exosmosis. Okay. So before moving to that, first we want to define what is osmosis. Osmosis is defined as movement of, clearly try to listen, a movement of water molecules from its higher concentration to lower concentration is called osmosis. So what is osmosis? Movement of only liquid, not solid, not gas. Movement of liquid from its higher concentration to the lower concentration is called osmosis. So there are two types of osmosis, endosmosis, exosmosis. Where this osmosis will happen? Osmosis will happen inside every plant, microorganisms, human beings, every animals. How movement of water will from one cell to another cells? Through osmosis only. So how it is happening? So we know the procedure, but little technical terms we want to see. So here first we'll see endosmosis definition. Osmotic entry of water into the cell or system. Osmotic entry of water into the cell or system is called endosmosis. So what is endosmosis? Water is getting inside the cell means that is endosmosis. Osmosis happening inside the cell, or osmosis happening that is into the cell means that is endosmosis. Next, exosmosis means osmotic withdrawal of water from cell or system. Water is moving out from the cell, or that will call it as, or coming out from the cell, that will call it as exosmosis. Clear, children? So there are only two types of osmosis, endosmosis and exosmosis. Endosmosis, endo means get inside. So water is getting inside the cell, that is endosmosis. And exosmosis, osmotic withdrawal. X means it is coming out of the cell. So or withdrawal of water from the cell is called exosmosis. Clear about what is osmosis? Moving of water molecule from its higher concentration to lower concentration is called osmosis. And here you want to remember, moving of water molecules from its higher concentration to the lower concentration through a semi-permeable membrane, or selectively permeable membrane or plasma membrane is called osmosis. Clear? So osmosis means it will happen in living system through a semi-permeable membrane or a selectively permeable membrane or a plasma membrane, moves from higher concentration to the lower concentration. It might be endosmosis, it might be exosmosis. Endosmosis means water is getting inside the cell. Exosmosis means water is coming outside from the cell. Clear, children? And here when we imagine, so I will explain with the help of two examples. Now take a seed, keep inside the water and leave it for 8 hours. And after 8 hours, if you locate the seeds, if you see the seeds, what will happen to the seeds? Seeds become swelled. And here what process has taken place? Whether it is endosmosis or exosmosis. What could be? Here water is getting inside the cell. So we'll call it as endosmosis. Clear? We'll call it as endosmosis. And you try it again. Take some apricots. Apricots means dry grapes. Take some dry grapes and put it in water. Leave it for 4 hours or 5 hours. After 5 hours, just observe the apricots or dry grapes. It will become swelled. What process has undergone here? Here again the dry grapes become swelled. Endosmosis has taken place. Why? The water was moving from higher concentration to the lower concentration. We know dry grapes there is no water, there is a water but it is very less amount, and outside when we are keeping water inside the bowl, no, that is only water in the higher concentration level. So here water is moving from higher concentration to the lower concentration. Higher concentration is your water and lower concentration is your water which is present in your dry grapes, and it is moving through a living membrane or moving through a membrane of a living substance. So that membrane will call it as semi-permeable membrane. So this is, we'll call it as osmosis. So example for endosmosis, you can try seed or you can try apricots, and you try a process for exosmosis also. Take a lemon, okay, take a lemon, cut it into small pieces, and these small pieces you just keep it inside some salt, okay. So you just take one bowl, keep more salt in that and add all chopped lemons, and you leave it for 8 hours. After 8 hours, you just open and see what will happen to the lemon. So here the salt doesn't contain any water or very low amount of water when we compare to the lemon. Lemon contains more water. So what is the process here? Water should move from higher concentration to lower concentration. Here higher concentration is lemon, higher concentration of water is present in lemon, and lower concentration of water is present in salt. So water is moving out from the cell. Here that's why we'll call it as exosmosis. Water is moving from the cell, from out of the cell, so we'll call it as exosmosis. So when you observe that lemon containing bowl, all it gets water, all you can able to see some water droplets or some water molecules will be in your bowl. So this process is called exosmosis. Whatever exa or endo, both move from higher concentration to lower concentration. When get inside the cell means endosmosis, get outside the cell means exosmosis. So this is the picture was given for exosmosis and endosmosis. So endosmosis, you just keep the water molecules containing bowl, your seed, all your waters are entering into your seed or apricot, so the seed becomes swelled. So this is a process of endosmosis. And come to exosmosis, I already told, keep some lemon pickle surrounding your salt. So here salt is very less. So Na plus, Cl, all your sodium chloride, your salt only, very less amount of water molecules comparing to the inner part. So inner part is your lemon containing more water when compared to the outer side. So from where the water will move? The water will move from your lemon to outside. So this process is called exosmosis. Clear, children? So what we saw? We saw two processes. What is osmosis? We saw endosmosis, we saw exosmosis. And you just practice in your home and how endosmosis will happening, how exosmosis will happening, and the definition is clear, and it is happening through a semi-permeable membrane. Clear, children? And next class we'll continue the one more type, very simple, one more type of solution, hypertonic, isotonic. That alone we'll see in next class. I think you are clear about the structure of microscope and what is osmosis, what are the types of osmosis, what is endosmosis, exosmosis. Please do it in your home, endosmosis and exosmosis. And next class I'll ask question based on this, and I'll continue the types of solution. With that, I'm going to finish the first chapter in biology of grade 9. Thank you, my dear grade 9.